ever try making a cake, right? But you just like chuck stuff in, flowers flying, sugar, eggs, everything, everywhere.
Yeah.
What are the chances you're going to end up with something edible, let alone delicious?
Right.
Probably pretty slim. Well, it turns out building the stuff of life, genes, and proteins, that might be even more complicated than baking without a recipe, which is kind of what you've sent us with this Part 3 ES.PDF material.
It's true. It really makes you appreciate the complexity. Even back in the 1960s, when scientists like Murray Eden at MIT were sort of wrapping their heads around, the math behind DNA and proteins, they were already asking some pretty tough questions.
Because it's not just about the ingredients, right? It's the order they go in. Like chocolate chips, great, but not if they're somehow inside the egg before you even crack it open.
Exactly. It's all about the sequence. And so you can, maybe try to, illustrate this with like a bicycle lock, right?
Yeah.
Simple one. 3 dials, 10 numbers each.
Yeah.
Not that hard to crack.
Yeah. But what if you had a lock and it had, hundreds of dials, each with 20 different settings, and you've got to get the right sequence?
No, my brain hurts a little bit. But okay, give me the protein version of this. Why are so many possible combinations an issue?
Yeah, because proteins, right, which are the workhorses of our cells, they're made up of these long chains of amino acids.
Mm-hmm.
And there are 20 different types. So you can think of like an alphabet with 20 letters.
Okay.
And the sequence of those letters determines how that protein folds up into its 3D shape, which then dictates its function. But even a short protein can have an astronomical number of possible sequences.
Okay, astronomical. Like more stars than we can see astronomical.
More stars than in every galaxy we know of combined. I mean, for a protein that's, say, 300 amino acids long, which is a common size, the number of possible combinations is 20 to the power of 300. That's more than the number of atoms in the known universe.
Okay.
And each protein needs a very specific sequence to work.
So finding a functional protein is like, what, searching for one specific grain of sand on every beach on Earth?
Yeah.
And then some. We're talking about that level.
You're getting a sense of the scale of the challenge. And this is what Robert Sauer, also at MIT, started really digging into in the late 1980s.
Yeah.
Because he was using this new gene editing tech that allowed him to actually measure how rare functional proteins are in that vast space of possibilities.
Okay, so what did he find? Because I'm already getting nervous. Was it as bad as everyone thought, this needle in a haystack situation?
It was even worse,
Yeah.
surprisingly. Sauer found that for a protein of, say, 100 amino acids, which is, even smaller than what we were talking about, only about 1 in every 10 to the power of 63 random sequences would actually fold into a stable, functional protein.
10 to the power of 63. I don't even know how many zeros that is, but that sounds really bad for the odds of just like random mutations hitting on something useful.
It is a mind-bogglingly large number. It's almost impossible to really wrap your head around. I mean, if you want to try and visualize it, imagine trying to find a single marked atom in the entire Milky Way galaxy.
Oh, wow.
Those are the odds we're talking about.
So evolution has to find that marked atom not just once, but for every single protein that makes life as we know it possible.
Yeah.
This is a high-stakes game of cosmic roulette. So if finding a functional protein is like winning some kind of cosmic lottery, how does evolution even get a foot in the door? It's not like it can just, buy a ticket and hope for the best, right?
Oh, it's a great question. And it kind of gets to the heart of two main scenarios that evolutionary biologists have proposed for how new genes might arise. It's like Richard Dawkins' Mount Improbable analogy. Have you heard of that one?
Yeah, vaguely. Refresh my memory.
Basically, that climbing a sheer cliff face straight up is, well, impossible.
Right.
But maybe, just maybe, there's a gradual slope on the other side of the mountain that evolution can climb, step by step by step.
Okay, so I'm picturing this Mount Improbable. What would be the first path then up this mountain of gene creation?
Okay, so the first path is what we might call peak-to-peak evolution. So imagine you've got an existing gene, it's doing its job, and over very long periods of time, it gradually mutates into a gene with a completely new function. Small changes upon small changes over generations.
Okay, so it's more of a slow and steady wins the race kind of approach.
Mm-hmm.
But didn't Sauer's research kind of throw a wrench into that with like those really, really slim odds of finding a functional protein just randomly?
Yeah, and you're right to be skeptical. I mean, remember how we were talking about proteins needing to fold into these very specific, really complex 3D shapes to function?
Yeah.
Well, Sauer's work showed that even small changes to a protein structure, like even just swapping out one amino acid for another, can make it unstable. It's more likely to fall apart if you make changes, basically.
Right. And a broken protein isn't going to help anything, let alone like push evolution forward. It's like trying to build a house of cards in a windstorm. One wrong move and whoosh, the whole thing collapses.
Perfect analogy. And that's where this idea of selection pressure comes in. If a mutation makes a gene worse at its job, the organism is less likely to survive, less likely to reproduce and pass on that not so helpful mutation. So evolution can't really favor changes that are detrimental, even if they maybe eventually could lead to something new down the line.
Okay, so much for the gradual slope. It's starting to sound like we're back to that sheer cliff face.
Right.
But wait, you said there were two paths, right?
I do.
What's the other option?
So the second path is kind of like starting in the valley and just hoping for a lucky break.
Mm-hmm.
This is the idea that new genes, they don't come from existing genes. They arise from sections of DNA that aren't currently doing anything supercritical, sometimes called junk DNA.
So they're just kind of hanging out.
In a way, yeah.
So these sections can mutate all they want without causing immediate harm because, they're not responsible for some vital function at that moment, like a genetic playground.
Exactly. It's like evolution can experiment in those areas without, breaking the whole system. And one way these genetic playgrounds might even arise is through something called gene duplication.
Okay.
So sometimes during cell division, a whole chunk of DNA just gets accidentally copied. So now you have two versions of the same gene, and that means one copy is free to just, accumulate mutations without impacting the function of the original.
So it's like having a backup file on your computer. Like you can mess around with the backup, and if you totally screw it up, no big deal, you still got the original. But even with this, like safer space to play around in, could those random mutations in these duplicated genes actually lead to something new and functional? Like, we're still dealing with that whole needle in a haystack improbability, right?
It's a huge question, yeah. Because even with a more forgiving environment to work with, that search space is still unbelievably vast, and those functional sequences are still incredibly rare. And, the problem gets even bigger when we think about the Cambrian explosion. We've just been talking about one protein at a time. But
right. The Cambrian explosion, that's when life on Earth went from, simple organisms to basically a full-blown underwater party, this massive explosion of new and way more complex life forms, seemingly out of nowhere.
Right. And it happened relatively quickly.
Yeah.
Geologically speaking, of course.
Relatively.
And so each of those new life forms would have required just a ridiculous amount of new biological information. I mean, new genes, new proteins, new cell types, all working together in a coordinated way. It wasn't just one lucky win in the cosmic lottery. It was like a winning streak that honestly defies imagination.
So we're talking about evolution hitting the jackpot, like not just once, but over and over and over again, and in a pretty short time frame, relatively speaking. Because didn't this Cambrian explosion, that didn't happen over billions of years, right? It was much quicker than that.
Yeah, that's one of the most, perplexing parts of this whole thing. I mean, the Cambrian explosion, it really was this just unprecedented burst of like biological innovation. Diversity and complexity of life just skyrocketing in, geologically speaking, a really short amount of time.
Yeah.
Like a few million years.
Okay, so a few million years, still a long time for us, but
On the geological timescale.
a blink of an eye.
Yeah, basically.
So we've got this massive increase in complexity happening really fast, and we're still trying to find that single marked atom in a whole galaxy, let alone like the thousands, maybe millions of marked atoms that you'd need to build these completely new life forms. It's like saying, yeah, sure, you could win the lottery every single day for a year, but also you have to juggle chainsaws blindfolded while you're dying it?
Yeah, I mean, you're not wrong to be a little skeptical here, because the sheer kind of improbability of it all, it's led some scientists to really question, like, can random mutations alone actually account for the Cambrian explosion? Because remember, natural selection, it can only work with what's already there. It's not like it can just create, new genes out of thin air.
Right.
It can only sort of favor those rare mutations that happen to be beneficial.
So it's like evolution is playing poker, but with a deck full of junkers, and it needs to draw a royal flush to win multiple times in a row.
Yeah, that's a good way to put it. And that's why this whole Cambrian explosion thing, it's such a fascinating challenge, I think, to how we understand evolution.
Yeah.
It forces us to ask, like,
Is there more to the story? Is there something else going on here?
Yeah, like what else could be at play? Are there
Yeah.
mechanisms beyond just pure randomness,
Yeah.
that might help explain how new genes, new proteins actually come about? I mean, we've talked about how massive that sea of possibilities is, right?
Right.
But are there like hidden currents within that sea that might be kind of steering evolution in a certain direction?
Yeah, I mean, that is the million-dollar question.
Yeah.
And it's something that, researchers are really actively investigating. And while we don't have, definitive answers yet, there are some really intriguing avenues of research that are being explored.
Okay, so like, what are some of the ideas that are on the table?
Well, one of them is this idea of exaptation.
Acceptation.
Yeah, it's a mouthful, right? But it's this idea that basically you have genes and proteins that already exist,
Mm-hmm.
and they evolved for one particular function,
Right.
but then they get repurposed for something completely different.
Okay.
So it's like, taking the engine out of a car
Yeah.
and using it to power a boat.
Uh-huh.
Instead.
Oh, I like that. It's like evolution is, recycling, repurposing,
Exactly.
working smarter, not harder.
Exactly.
Yeah.
So another really interesting area is this whole world of horizontal gene transfer.
Mm.
And this is where organisms, particularly single-celled organisms, can actually acquire genes from other organisms around them.
Wait, hold on. So you're saying it's not just about like slowly accumulating these mutations over generations. There might be these like shortcuts, these like genetic swaps happening between organisms.
Yeah. And it's honestly more common than you might think, especially in like the microbial world.
Mhm.
And this could be, a really significant driver of how new things come about, because it allows organisms to kind of acquire these pre-existing genes and functions in a way faster way than having to evolve them from scratch.
See, that's what I'm talking about. So it's not just about finding that one marked atom anymore. Maybe there's a whole treasure chest of marked atoms out there just waiting for something to come along, pick them up, and put them to use in a new, even more creative way.
Yeah, that's a great analogy. And then there's, even more to consider. Like we've talked about junk DNA, right?
Yeah.
And how scientists used to think it was just that junk. But we're learning more and more that these non-coding regions, they actually might play a really important role in regulating how and when genes are expressed,
Okay.
And that can have a huge impact on evolution too.
It's like we thought we were just looking at the tip of the iceberg this whole time, but it turns out there's this whole massive, intricate structure underneath the surface that's influencing everything.
Yeah, it really makes you appreciate how much more there is to learn about, just this incredible story of life on Earth.
And how it got to be so complex.
Exactly. And how it got to be so complex. We're just starting to, scratch the surface. And I think the more we learn, the more we realize that kind of simplistic view of evolution that we often hear, random mutations plus natural selection. And it really is simplistic. The reality is way more complex, more nuanced, and honestly kind of awe-inspiring, don't you think?
Absolutely awe-inspiring. I mean, we've gone from baking a cake to the vastness of outer space, and now, like, hidden genetic treasure chests, all thanks to your awesome questions and, of course, the material that you sent us.
Well, it's been a pleasure, as always, diving into this with you.
And to you, dear listeners, we leave you with this thought. If random mutations aren't the whole story, what other forces might be shaping the incredible tapestry of life? What other mysteries are waiting to be unraveled in that vast, dynamic code we call DNA? Keep those questions coming, deep divers, and until next time, keep exploring the